agenda. Next up is Lexi Sumner from Fisheries and Oceans Canada to talk to us about threats and opportunities related to larval assessment. Thank you, Lexi.
Hello, everyone. My name is Lexi Sumner. I'm a larval assessment biologist with Fisheries and Oceans Canada. I'm presenting these slides that were put together by other larval assessment biologists at Ludington and Marquette, and also Nick Johnson from USGS. So a quick overview of what we're going to talk about today. You've heard a little bit about what larval assessment is, but I'll go through the different tools that we use to capture larval sea lamprey to inform treatments, and talk about why it's an important part of the program. And then we'll go through some of the threats that we see coming up these days, as well as some opportunities. Some of the threats include how we decide which streams to treat, climate change and how that impacts our ability to capture lamprey, issues with accessing streams to survey. And then some opportunities include some research projects that are ongoing that provide some exciting opportunities for future ways to collect larval lamprey. So larval assessment, this is the part of the program that I work in. I lead a small crew that travels around Lake Ontario and Lake Erie tributaries to capture larval sea lamprey. The information that we gather from these surveys is where the lamprey are. Where the lamprey are, distributions within stream and which streams harbor sea lamprey, and also some information about the size of the lamprey in the streams and the density, how many there are there. We have three main tools that we use. Primarily, we use backpack electrofishing, where we use low voltages of electricity to irritate lamprey out of the sediments where they're burrowed so that we can scoop them up. In waters that are too deep or too turbid to effectively backpack electrofish, we use a type of lampricide. Bruce explained a little bit the lampricide that we use. It's granular balicide, and we apply that to the surface of the water. They're grains of sand that are coated in niclosamide. They settle to the bottom of the water column, and the active ingredient is released at the sediment where the lamprey are. And similarly, they're irritated out of the burrows where they'll swim up to the surface and we can scoop them up. Finally, we have the deep water electrofisher to capture sea lamprey in deep waters, and this is mainly used on the St. Mary's River, So in addition to doing surveys to capture larval lamprey, we also do habitat surveys so that we can understand how much prime larval habitat there are in each of the streams that have sea lamprey. And if we couple the results from our habitat surveys with the density information that we gain from our larval assessments, we can gain insight into the total stream population of lamprey in each of the lamprey producing streams. So why is larval assessment important? Sea lamprey control is a data-driven program. Data collected through larval assessments directly support the control decisions, like where to treat, why we're treating, and when we should treat. Without reliable larval assessment data, the control program wouldn't be able to proceed. So we find the streams that harbor sea lamprey, larval sea lamprey, and then we also prioritize which streams should be treated. We can't treat every stream that has lamprey in it in each given year, so we have to rank the streams for treatment so that we get the most bang for our buck. Following treatment, we'll return to streams that were recently treated so that we can determine whether or not that treatment was effective. So we'll use the same tools, backpack electrofishing or granular balicide, to see if there's lamprey that survived the treatment. And then the final thing that we do that helps support treatment planning is finding where in the stream we should apply chemical from. We don't want to miss any lamprey in the stream, so we want to know how far up they get each of the tributaries, but we also don't want to unnecessarily treat parts of the river that don't actually have sea lamprey in them. So we'll do surveys up each tributary to find out where we stop capturing larval lamprey. So we'll go through some of the threats that we see in the program. Primarily, are we treating the right streams? There's we've seen a little bit of a mismatch in some areas between where we're catching larval lamprey and the adult measures that we see with the adult index. So in some areas we're having trouble finding large lamprey that should prompt a treatment. However, the adult index in the lake still remains above our target levels. And it's increasingly difficult to find those older lamprey in the streams. So even though sometimes it's hard to catch lamprey in the streams, we'll still see adults running up the river to spawn as well. Another threat is staff turnover at the control agencies, DFO and Fish and Wildlife Service, and also the partner agencies. So a loss of experienced technicians and biologists who have years of experience with the program. And also, are the lamprey surviving treatment more, what Aaron just presented? Climate change is a big threat. With increased water temperatures, sea lamprey will grow faster, which might necessitate streams be treated more frequently. Streams that were previously too cold to support a larval population may be warming up, and so those lamprey can infest new tributaries where in the past they didn't survive. Similarly, increased length of infested stream, how far up the lamprey are going up the river. This requires more survey effort for us to find the lamprey, and consequently more treatment effort to treat those higher distributions. Flooding can be a big issue. Flooding and high water can make assessment work difficult and unreliable. Sometimes we can't safely access the stream after a big flood event. And even if we can get in there, there's probably a high likelihood that we can't observe the lamprey that are irritated to capture them. And also an increased occurrence of barriers being breached so that spawning lamprey can get above those sea lamprey barriers and infest areas of the stream that might be harder to effectively treat. Similar to what Bruce was explaining earlier, getting permission to access lands to do surveys. We do our surveys in public lands and parks, both state and provincial lands, tribal and First Nation territories, and we have to ask to access people's private land. So if people aren't willing to have government staff access the lands, or specifically sea lamprey control staff, if they're not supportive of the program. Species at risk is also an issue, particularly when we are using granular balicide to survey for larval lamprey. As was stated earlier, balicide was developed to be a molluscicide, and so in areas where there's at-risk mussel species, it can be harder to gain permits to do our assessment work in those waters. And then a big one for the program overall is the loss of social license and increased public sensitivity to lamprey use in general. So even if we are not using lampricides when we're doing the surveys, sometimes folks won't want us to survey their streams because if we do catch lamprey, that could prompt lampricide treatment on the rivers going through their property. And one of the problems with the success of the program is that there's diminishing public recognition of the importance of sea lamprey control. If people don't see the negative impacts of lamprey predation on native fishes, they might not know how important the work is. But all that to say, we have a lot of exciting opportunities in the program, and here are some ways that we can make the good sea lamprey work that we do even better. We hope to do some cross-training for staff, both within each of our individual offices and across the different agent offices between Fish and Wildlife and DFO. The adoption of new technologies. We've made some failures in the program and learning from them so that we continue to focus on improving our ability to detect and quantify lamprey. We've been collecting larval data for decades, and so we have vast data sets that we could use to answer some big research questions. Further refinement of new and emerging tech that we've been dabbling in, and I'll be going through some of those new opportunities in the following slides. And then also engagement with researchers, working with them directly to help develop and refine the research questions and the equipment and models that we can use to answer those questions. So one research program that I've been a part of a few years ago was using environmental DNA, or eDNA, to detect sea lamprey genetic material in streams. So if we're sloughing off genetic material in the water, presumably those pieces of DNA can be sampled just through water sampling. So we did a 2-year research project where we collected eDNA samples and paired it with our conventional electrofishing surveys to see if the results of eDNA aligned with what we were seeing in our electrofishing surveys. And for the most part, there was really good alignment in what the results of the study showed, and in some situations, eDNA was more sensitive, not surprisingly, than electrofishing, where we would catch novel detections of eDNA when we weren't capturing actual individual larvae in our samples. There's a lot of opportunities where we could use eDNA. It could be used for detecting sea lamprey in streams that have never harbored sea lamprey before. So instead of putting a huge amount of effort into electrofishing large stretches of river just to say that we didn't catch anything, we could more efficiently take water samples, and then if sea lamprey genetic material is detected, then we can better plan our surveys to try to capture those individuals. Normally for our electrofishing work, we finish late September, early October, but we found that we could use eDNA approaches further into the winter, so it could expand our sampling season. And then there's some promising, there's some hopes that you could perhaps use eDNA to quantify larval populations. To make any decisions about treatment, we would have to capture the lamprey and measure it, but it would be nice to see if there's more densely populated streams. Moving on to the SLAP program, the Sea Lamprey Aquaculture and Procurement Program, led by John Hume. This program allows us to better understand larval growth, survival, sex, and transformation. In this program, they are planning to complete the life cycle of a sea lamprey in lab settings. And the lab experiments have revealed insights into the ecology, physiology, and genetics of larvae. And in the following slide, I'll give you a little case study of what the early things that have been learned with this program. So one of the questions was, how does density influence growth and survival? So if you look at these larval sea lamprey, I'd like you to consider, as we're going through this, what the numbers on the orange tape might reflect. So we'll go through the average length of these amicetes at 9 months of age, and the survival of these amicetes, of these larvae. So all of these lamprey were spawned from the same day by the same parent. The only difference was how densely populated each of their study tanks were. So there's pretty similar rates of survival, but the growth rates exhibited by these lamprey are pretty different. Those that were more densely packed into a tank grew a lot more slowly, and those that were sparsely populated in the tanks had a lot faster growth rates. And this has implications for when the lamprey will transform into their parasitic phase of life. Another question that SLAP has been able to answer is, will lamprey grow faster in warmer streams? So they used temperature-controlled rack systems to test 3 temperatures, and they found that lamprey grow faster when temperatures are warmer. And this is important for the control program. As we said before, if river temperatures are warmer, lamprey will be growing faster, like we're seeing through the warming of our waters. And then finally, we'll move on to an opportunity through the Supplemental Control Program led by Nick Johnson at USGS, where they're able to get better estimates of reproductive contribution, larval growth, and survival through pedigree assessment of wild lamprey populations. Pedigree reconstruction, in simple terms, is determining if larvae collected from a stream are brothers and sisters. If they're brothers or sisters, they're the same age and are born the same year by the same parents. And by reconstructing during the pedigree, you can estimate how many adults produced year classes of larvae. If you recapture a family member in a future year through close kin mark recapture, you can estimate the family-specific growth, survival, and drift downstream. So in this figure, you'll see the dots they're flanking the sides of each of these figures are the individual adult lamprey, and the lines are the larval lamprey produced by those adults. So you can see in some systems, there's many adult lamprey that are contributing to that larval population. Elsewhere, there might be one or two adults that are contributing to the larval population. So in summary, larval assessment is an essential part of the sea lamprey control program. The work that we do helps inform when, where, and why we treat rivers. And we need to acknowledge the threats that we have so that we can take advantage of the many opportunities that we have as a program. Thank you very much, and I'll welcome any questions if we have time.